Okay, so welcome to this session. Basically, for this session, we're going to be looking at the molecules known as the proteins. So we're going to be looking at proteins. We're going to understand that actually proteins are the cell machineries. They are the machinery of life. They are the machineries of the cell. And by definition, a cell is a basic structural and functional unit. It's a basic structural and functional unit of an organism. So proteins are the machinery of life, and we want to understand why they are the machinery of life. These are some of the lesson questions that we're going to consider. Why is it that proteins are important in living organisms? I'm also going to answer the question, what is a protein's conformation or configuration, and why is it important? What is a protein's conformation and why is it important? Furthermore, you're going to ask the question of what the monomers of proteins are. And then we're going to answer the question why there are so many kinds of proteins. And then we're going to have an overview or description of an amino acid. How is an amino acid? What's the structure? And what's the importance of such a structure? And then we are also going to discuss what makes one amino acid to be different from another. And then we're going to also answer the question, if time will allow, how peptide bonds form. And then by the time we reach at this point, time would have been up. So most likely we may end on what makes one kind of amino acid different from another. So the first question that we need to answer is, why are proteins so important to life? We need to understand that proteins are actually complex organic compounds, and they contain carbon, hydrogen, oxygen, as well as nitrogen. And in some cases, we have sulfur being present in proteins. So we have some amino acids that contain sulfur. So these are complex. They are actually the most diverse functionally and chemically biological molecules that we have. They are the most diverse. But why are they so important? So like I said in the preamble, I said that proteins are the machinery of life. How are they the machinery of life? How are they the machinery of life? So they are the machinery of life because of this process. I'm sure you may have looked at this process. This diagram is simply describing what is known as the central dogma of molecular biology. Central dogma of molecular biology. And it starts with DNA, which is the information storage molecule. And this DNA codes for information that is essential for the synthesis of proteins. DNA codes for information essential for synthesis of proteins. So DNA is transcribed. The antisense strand of DNA is transcribed, leading to the synthesis of formation of messenger RNA, which is the information carrier. It acts as a messenger. And messenger RNA is involved in the process of translation, translated into amino acid chains. So remember, the process of translation will require the transfer RNA being involved. The transfer RNA containing the anticodons forming bonds with the codons found on messenger RNA. We have the ribosomal RNA forming what are known as the ribosomes. And through that process of translation, we find amino acid chains being formed. And amino acids are actually the monomers for proteins. And these proteins now act as the active cell machinery, the active cell machinery. No wonder they're saying that proteins are very important because they are the machinery of life. They are used for various functions. Proteins act as catalysts. Proteins act as supporters. So they have a support or a structural role. So we have protein fibers that act as structural roles. So we have proteins such as keratin, which is found in hair. We have fibrin, which is found in blood, especially it's involved in blood clotting. We also have collagen. Which is found also in which forms a matrix of skin and ligaments as well, and tendons. So we have these proteins acting as supporters. And we see that proteins can be assembled into various shapes, and this allows them to serve as major structural components of cells and tissue. That's the more reason why it is commonly known that the proteins are important for growth and repair because they act as structural support. Remember, when we hear about carbohydrates, we hear, no, these are energy-giving foods. When you hear about proteins, you say, no, these are used for growth and repair. It is for this reason, structural support, that they are used for growth and repair. So they are used as supporters. They are used as catalysts, and these biological catalysts are known as enzymes. So these facilitate or speed up the chemical reaction by lowering the activation energy of a reaction. That will become more clearer when we look at enzymes themselves as a topic on its own. Enzymes also act as receptors and communicators. They act as receptors and communicators. So proteins rather act as receptors and communicators. They are able to enable different cells to be able to communicate together. They're able to enable different cells to be able to communicate together. And then we find that proteins are also important as they act as transporters. So we have a variety of we have a variety of proteins that act as transporters. Of importance to mention is the protein hemoglobin, abbreviated as HB. So this transports oxygen in the blood. We also have a lot of other membrane transport proteins which help move ions and molecules across cell membranes. So they act as transporters. The other function is that they act as movers, or they have a function in motion or movement. In this case, an example are the muscles. The muscles contain two kinds of protein filaments. So they contain actin and myosin. These are proteins. And this enables a person to move from one place to another. For us to move, it's because the muscles need to contract. And for those muscles to contract, there should be a sliding effect between actin and myosin. So they act as movers, or they're involved in motion. Why are proteins so important to living organisms? They act as defenders. They act as defenders. So we have proteins that are able to use their shape and recognize foreign bodies, foreign organisms, foreign microbes, even cancer cells. So these form the core of what is known as the endocrine and the immune system to defend our bodies against diseases, against microbes, against any foreign body that comes in. So they're very important. They act as defenders. The other function is that enzyme proteins rather act as storage molecules. So for example, calcium, when we look at the elements calcium and iron, these are stored in the body by binding as ions to storage proteins. So they act as storage materials. So proteins act as supporters. No wonder they are important for growth and repair. They act as enzymes, receptors, and communicators, transporters, movers, defenders. Very important functions. The other question we need to ask is, why is it that there are so many different kinds of proteins? There are actually so many different kinds of proteins. Now, the reason is very simple. The reason is very simple. It's because there are different amino acids that we find in nature. The different amino acids that we find in nature. And generally, commonly, we have 20 different amino acids in nature. What's the implication of this? It means that if someone has, or rather, if there is a protein with just one amino acid, it means just from one amino acid, there can be 20 different proteins that can result from just one amino acid. In short, we are saying for us to know the number of proteins that can result from a certain number of amino acids, we put a certain power to the power 20. So we power 20 to a certain number. If we have one amino acid, so we're going to say 20 to the power 1. It means that one amino acid can give us 20 different proteins. It means if we have 2 amino acids, we're going to say 20 to the power 2. And this is going to give us 400 proteins. So 2 amino acids will give us 20 to the power 2 different proteins. What if there are 3 amino acids? It means that it is going to be 20 to the power 3, giving us 8,000 different proteins just from 3 amino acids. If there are 4, it means they are going to give us 20 to the power 4, and that's 160,000 different proteins. Why are there so many different kinds of proteins? Now, we can see that just a small chain of amino acids, a small chain of 4 amino acids can produce a very huge number of different combinations. Just 4 amino acids giving us 160,000. Wow, that's a huge number. Now, when we look at the proteins that are found in living organisms, generally they have about 500 amino acids. 500 amino acids. Now that's 20 to the power 500 different proteins. If you are to punch that in your scientific calculator, most of these calculators, they will just bring an error. They can't even compute that number. Most of these scientific calculators, normal scientific calculators, you punch in 20 to the power 500. They're going to give you a math error because that's a very huge number. It's a huge number. So no wonder we find that proteins are actually the most diverse and the most sophisticated molecules that are there. So that was just a preamble. That's the importance of proteins. We need proteins. They are very important. They are very important. So what is a protein conformation? Remember, during the process, post-translational process, one of the modifications that we look at is the protein shape. The protein need to be shaped in a certain configuration, is in a certain conformation. It need to be in a 3D conformation, a 3D shape. Now, for it to function properly, that protein needs that shape. So in short, what's a protein's conformation? Simply its 3D structure. It's 3D structure. So it needs to be forwarded using molecules that are known as the molecular chaperone. The molecular chaperone assists in the shaping or in the bringing of this protein into a shape. Now, why is this protein's conformation important? It is very, very important. The shape is very important in that it is essential to its function. The shape of the protein is essential to its function. And I'll show an example of a familiar protein that we may know. And every protein is adapted for a job. Remember, every protein is adapted for a job. For example, we mentioned hemoglobin. Hemoglobin is adapted for the job of transporting oxygen in red blood cells. So it means that this hemoglobin needs a certain conformation, a certain structure for it to be able to transport oxygen efficiently. So every protein is adapted for a job. The other point to note is that actually form follows function. Form follows function. The shape follows the function. The form of the protein follows the function, what the protein does. So the shape of the protein is essential to its function. Every protein is adapted for a job or function, and form follows function. Remember, enzymes, we know enzymes, biological catalysts. Enzymes have an active site. So we have, for example, the enzyme sucrase, which is used in the hydrolysis of sucrose or breakdown of sucrose to glucose and fructose. Now, sucrose is actually, if you can remember from your carbohydrates, is a disaccharide made of glucose and fructose. And so we have this sucrose enzyme with an active site. Now, that active site, the substrate of sucrose need to bind to the enzyme at the active site. Now, if that active site, if the conformation of the enzyme is disrupted, this sucrose will not bind to the enzyme at the active site. Hence, we find that this process of hydrolysis, this process of digestion, this process of breakdown of sucrose will not take place because the shape of this enzyme has been disrupted. So this is an example of why the shape of a protein is important. It's very cardinal. Are we together so far?
Yes.
Yes, sir.
Okay, great. So what are the monomers of proteins? I think I preempted the answer to this question. I did mention when I was explaining the central dogma of molecular biology that during the process of translation, we have a polypeptide chain, which is made of monomers known as amino acids. So what are the monomers of proteins? They are amino acids. Now, what's the structure of an amino acid? This is the structure of an amino acid. This is the structure of an amino acid. When we look at the name amino acid means that it has an acid component, an acidic component, and an amine or amino component. Now, there are different functional groups in nature. We have hydroxyl functional groups, which are found in all these biological molecules? Okay, someone raise their hands. You can speak.
Sir, I remember you mentioning something about a
Yes,
molecule.
but there was
Okay, the lady first.
I remember you saying something about a molecule that assists in the shaping of a protein. I didn't get the name of the molecule.
It's a chaperone.
All right, thank you.
Yes, chaperone. Yes. Okay, thank you. Welcome. Sir, you can ask your hand. You can ask your question rather. You had your hand up. The other person that had the
so
question.
yes, the part where you said that enzymes are specific. So does it mean that I mean proteins are specific as well? So does it mean a particular protein can only work with what is a specific function only? For example, hemoglobin can't perform another function apart from carrying oxygen.
Yes, that's what it means. We have an enzyme, for example, I gave an example of an enzyme called sucrase, which hydrolyzes sucrose. It means that it cannot hydrolyze maltose. So they are specific. They have a special function. Hemoglobin its function is to transport oxygen, and that's what it does. Yeah, so that's what it means. We're okay? I've answered the question. Okay, then we continue. So saying amino acid, it means that it has an acidic component and an amino component. So when we look at an amino acid, an amino acid, it is called amino because it has an amino group, and it is called acid because it has a carboxyl functional group. I was saying that there are different functional groups in nature. We have hydroxyl functional groups. These are common in all these molecules. These are the OH functional groups. You find them in carbohydrates, in proteins, in lipids, in nucleic acid. They are all over. Those are hydroxyl functional groups. You have the carbonyl functional groups, which is really made up of two types of functional groups: the aldehyde functional group and the ketone functional groups. These are found in carbohydrate mostly, but as we are going to see in proteins, after the formation of peptide bonds, the carboxyl functional group, this carboxyl group of the protein is converted into a carbonyl group. So I have different functional groups in nature. We also have the sulfhydryl functional group, which is found in proteins. Certain amino acids, as we're going to see, contain sulfur, and sulfur is important for forming those sulfhydryl functional groups. We also have the phosphate groups, which, if we can remember from nucleic acids, are the ones that are responsible for making DNA to be acidic. So those numerous phosphate groups on DNA makes it to be acidic. So in amino acids, we have the amino group and the carboxyl group. Now, all amino acids have the same central structure. They have a same or common core structure containing a central carbon, asymmetric carbon. This is an asymmetric carbon, and it is known as alpha carbon. It's asymmetric. Remember, an asymmetric carbon is a carbon which is bonded to 4 different atoms or groups of atoms. Asymmetric carbon. Okay. So this is the carbon known as the alpha carbon. So generally, each amino acid is actually, each amino acid has this common structure. It has this common structure. So I will avoid those rough markings. Yeah, so it has this common structure. However, they are going to differ on the R side chain, and we're going to look at that later. So in short, we are saying the structure of an amino acid is it has an amino functional group, NH2, it has a hydrogen atom, and it has an it has a carboxylic acid group and a distinct ARA group. Now, this is very distinct because it's different from all amino acids. Now, the amino acids in nature usually, or rather at neutral pH or in solution, occur, exist, they do not exist as this, they exist as dipolar ions. They exist as dipolar ions. So they exist as dipolar ions in that they will have the negative, this one becomes negative charged, this one becomes positively charged. So this carboxylic functional group acts as an acid. So it is able to donate. It's able to donate a proton, a hydrogen ion. It's able to donate a proton, while the amino functional group is able to accept the proton. Hence, this one becomes protonated. This one becomes a negative charge. This one becomes positively charged. And amino acid exists in living cells in solution at pH, neutral pH, as dipolar ions. They possess a positive charge and at one end and a negative charge at the opposite end. So this is generally how amino acids actually exist in the cell pH. This is how they exist in the cell pH. Now, these charges that are found on these functional groups are actually very important. One, they help the amino acids to be able to stay in solution. And when they are in solution, they are able to interact with one another and also with different other atoms. That's the first reason. It helps the amino acids to be able to stay in solution and interact with one another and other atoms. The other reason is that they affect how the amino acids react chemically. So it affects the amino acids chemical reactivity. Now we see that I've explained that these amino acids have these amino and the carboxyl groups, which are able to accept and to release protons, as I have explained, leading to the formation of these dipolar ions. Hence, this ability, this property of them being able to donate and receive protons makes amino acids to be able to resist changes in the pH. They resist changes in the pH, in the acidity and alkalinity. Therefore, they tend to act as buffers, biological buffers due to that. Now, what makes one amino acid different from another? I've already mentioned that the big difference between amino acids is the ARA group or the variable group. The ARA group or the variable group or the side chain. So ARA group, variable group, side chain. This makes different amino acids to be different. So each amino acid is exactly the same, as I said. It has a common core structure, but they're going to be different in the ARA group. So that little side chain, is the one that makes one amino acid to be different from another. So for example, the simplest amino acid that we have is glycine, in which the ARA group is simply a hydrogen, And we have other amino acids such as alanine, in which the ARA group is actually a methyl group or CH3. So we have about 20 standard amino acids. 20 standard amino acids. So on this table, remember I mentioned that the unique character of each amino acid is determined by the nature of the ARA group. The nature of the ARA group determines the unique nature of the amino acid. So we see that unless actually the R group is a hydrogen atom, as in unless it is a hydrogen atom, as in the case of glycine, amino acids are actually chiral. They are chiral. Apart from this one, amino acids are actually chiral and can actually exist as two enantiomeric forms as D and L. However, in the system, living systems, what we find is that there are only L amino acids that are found in proteins. So the D amino acids are actually rare in nature. So the R group, the variable group, determines the chemistry of amino acids. For example, the amino acid serine. For example, the amino acid serine. This amino acid serine has the R group as CH2O2. This makes this molecule to be a polar molecule. So serine is a polar molecule due to this side chain of OH, CH2OH. This side chain makes it to be a polar molecule. In contrast, when we look at alanine, alanine has the methyl group as its side chain. This makes alanine to be a non-polar amino acid. So we find that these amino acids properties are determined by their side chain. Now, most amino acids are actually neutral because they have one carboxylic and one amino group. When you look at acidic amino acids, usually they are going to have two or more carboxylic functional groups, or basic amino acids will have two or more amino groups. So among these 20 amino acids, note the following, that we have two amino acids that are sulfur containing. Note the following, we have two amino acids that are sulfur containing, and we can see them here. The sulfur containing amino acids, we have methionine, sulfur containing methionine here. We have methionine there, and we have cysteine. These are special amino acids. They are sulfur containing amino acids, methionine and cysteine. The other thing that we need to note is that we have glycine, we have arginine and histidine, which are basic amino acids. We have aspartic acid and glutamic acid. Aspartic acid and glutamic acid, these are acidic amino acids. And then we have proline. Proline is an alpha amino acid. And then those that are aromatic, these contain rings, as we have seen, these aromatic rings. So these are amino acids. These are examples of amino acids, the 20 common amino acids. Okay. Do we have any questions? Hello, any questions? Or is it clear?
So I have a question.
Yes.
When you place an amino acid in a pH,
I'm unable to get you.
in a, I'm